Hello, yeah, I'm John. So I'm a postdoc here at Oxford as well. I'm starting today with this idea of all the plastic that we throw into the ocean. And you've probably heard about this quite a lot in the past, that there's plastic everywhere. Almost everywhere you can find it, we find these lumps of plastic waste. You might have even heard that there's these microscopic fragments of plastic called microplastics that you find inside the tissues of animals and even inside our own bodies. And we don't know what the sort of long-term ecological effects of all the plastic we find out there is going to be. So clearly we need to have some sort of solution for the way that we deal with plastics and this type of material and things. But when we don't want to get rid of it altogether, it's really useful. We can make lots of useful things out of plastics, like waterproof jackets and life-saving water filters and bandages and things like this. So what if there was a way to make plastics that was from renewable sources and that was biodegradable as well? So that's where bacteria come in. So I'm a microbial synthetic biologist. My favourite type of microbe to work with are bacteria, and these are single-celled microscopic organisms, and some that you might have heard of are Escherichia coli, Listeria monocytogenes, Salmonella typhi, Vibrio cholerae, and some of these are quite sort of scary, nasty sounding ones with a pretty bad reputation, but I want to try and remediate a bit of that today, because there's some really interesting, cool things that bacteria can do as well. Some bacteria are able to make stuff, are able to make biomaterials. Biomaterials are pretty funky and can be pretty futuristic looking, but really biomaterials are just materials that are made from some sort of biological process, like wood or wool or something, count to be biomaterials as well. And these are some sort of the more futuristic and modern things that people are making out of biomaterials now. This is a dress made out of a seaweed leather, or a leather made out of a seaweed polysaccharide. This is a hat that's made out of synthetic spider silk. So there's a company called Bolt Threads that genetically modify bacteria to make synthetic spider silk, and they made this cool hat out of it, obviously. These are tiles, like you might find in the bathroom or something like that, but they've been made from a biological cementation procedure using bacteria as well. And this is a face mask made from a bacterial type of leather as well. So lots of futuristic, cool things that you can make out of bacteria, or you can make with bacteria. But what about plastics? We actually find plastics, quite commonly in nature. There's a common example in the leaf cuticles in plants. They have this cuticle that's made out of a polyester. You know what polyester is? This is the nylon and things like this, right? And plants use this polyester that's on their cuticle like a little raincoat. But bacteria can make plastics as well. This in the background is an electron micrograph of a bacterium called Cupriaridis necator. I'll talk a little bit more about that later. This is a bacterium that can make a plastic called polyhydroxybutyrate, PHB. And all these big white blobs that you can see inside the cells are granules of this plastic material. They use the word blob on purpose because they use it in the same way as we use fats and stuff. They use it as a way to store energy away for the future. Cysts are completely biodegradable plastic because the bacteria make it so that they can eat it again later. And there's lots of bacteria that live in the soil and things that both produce this plastic and also eat it. So it's completely biodegradable plastic. It doesn't produce, or they won't produce these microplastics and things too. What if we could then produce that from renewable sources and also produce enough of it to make some cool products out of? So that's where synthetic biology comes in this project. So synthetic biology is when you put together new DNA sequences, or put together DNA sequences together in different ways to make new biological functions. And it's a bit like writing software on the computer. So you might write a software program that says, if the location is the old fire station, then John should do his talk. And if the talk is good, then hopefully you'll all applaud. And if the talk is not any good, then I'll go back and practice it somewhat. And the computer interprets this as the big string ones and zeros and executes the function that you've read. And we can make this similarly in DNA as well. So a common type of this sort of if statement here that's a sort of logical, when should I execute the function, is this thing called a promoter. And this is a sequence of DNA that says, when should I execute the function, and sort of how loudly and how much should I execute the function. And the example that I have here is this really common gene that we use a lot of time called green fluorescent protein. So this is instructions on how to make a protein that glows bright green. And we write this in like I mentioned, in this string of A, T, C's and G's that make up DNA, and we install this into bacteria, and we have these beautiful green fluorescent bacterial colonies, which are really cute to work with. And In our lab, we make these really interesting genetic programs called that operate at this optogenetic level. And these programs, optogenetics are really sophisticated, complicated genetic programs that allow bacteria to respond to specific light signals. So you might encode a program that allows the bacteria to begin executing a function when it senses a green light, and if you turn on the red light, you may stop executing that function. And we use these cool bioreactors, these little robotic bioreactors. I brought one here so you can see how small and funky they are. These were invented by my boss a couple of years ago to do this. And that's what it looks like inside when we use optogenetic functions, where it becomes like a little bacterial disco on the inside. And I say that they're robotic because the bioreactor can measure lots and lots of different things about what the bacteria are doing. It can measure these fluorescent signals, and if we're using green fluorescent protein or something like that. It might measure how fast they're growing or other properties about what the bacteria behaviour is. Can then make decisions about what to feed the bacteria or if to feed them at all, or what temperature to grow the bacteria at, things like this. I say behaviour on purpose because when we use optogenetic programs, we often find that bacteria don't respond very well. Their behaviour changes in ways that isn't useful to us, and obviously it's not useful for them either. They start growing slower and things. This is because of this concept of genetic burden, where too many genetic modifications can be harmful for bacteria. And this is because bacteria are really, really efficient, sophisticated systems. They've evolved through natural selection to be as efficient as possible and waste as little as possible. And we find them throughout, everywhere throughout nature. We find loads of different bacteria, find loads of interesting, cool things that we can do. You might find a cool new one and think, oh, I want to make that, make that useful. I want to do something cool with that, make that into a technology. So you might decide then to make a genetic modification to enhance that property or make it be able to do something slightly different. So you have then this bacterium that's maybe got cool new abilities. In this case, it's been given the ability to ride a bicycle. You can zoom around town. But you might want to enhance that ability even more and sort of make it even more sophisticated. So you make you encode lots more complicated genetic programs at the DNA level. And it becomes a bit like trying to ride a bike with a big tool bag on your back that's really, really heavy. You get tired out really, really quickly. So you might have all the equipment and all the tools do some really cool, sophisticated stuff, but no energy to actually carry any of that out. And the way that we might think about, or might respond to this is by thinking about microbial communities. A microbial community you might be familiar with is in kombucha. So kombucha is a tea drink that's made by a fermentation carried out by a community of yeast and bacteria. So they work together to make this tasty tea drink. And in my research, I work with synthetic bacterial communities. So we bring together different bacterial strains with a small number of genetic modifications each. So you have a variety of different bacterial strains, each that have been genetically modified in a very small and subtle way. And that allows them to work together in this sort of more complex orchestrated process. And it becomes a bit like trying to design an ecosystem from scratch, where you might have woodlice and worms and things in the soil that enrich the soil that allow plants and trees to grow, and these form a habitat for squirrels and birds and things like that. But of course, one of the communities that I work with in the lab is contains this bacterium Cuprialis nectator that I mentioned earlier that produces these plastics. We'd like to make these plastics from renewable sources, and our super renewable source of sugar is from sugarcane. This contains this sugar called sucrose. Cuprialis nectator, though, is unfortunately not able to eat that sucrose. It can't turn it into plastic. But if we grow it in a community with this other bacterium called Bacillus subtilis, that bacterium is able to convert sucrose into glucose and fructose. So the bacillus takes the glucose, yummy glucose, kind of eat lots of that, and the Cuprialis goes, oh, thank goodness for that. Someone's turned that sucrose into some fructose for me, grab that fructose, turn it into some plastic. But what if, though, one of those bacteria outcompetes the other one? What if one bacterium becomes really dominant and we don't have any plastic anymore, or the other plastic-producing bacteria is not able to grow anymore? Well, that's where these little robotic bioreactors come in. They can measure the composition of the community. They can measure the properties of the culture, the two different bacteria that are in there, and make predictions about how much of each different bacteria are present. And then we can decide how to feed the bacteria, how to treat the community in there to return the composition of the community to something that produces lots of plastic for us. So how much plastic can we actually make from a process like this? Well, I want to screw about half a litre of bacteria. That's about a pint's worth, if you're fancying one like I am. And only produced about this much plastic. So that's a photograph of the plastic that's in this tube here. So it's all just in the bottom of this wee tube. That's 44 milligrams worth. So it's not very much. So we need to make it a bit better than that. We want more plastic than that. And the way we think about that is by going to synthetic biology and thinking about how to genetically modify these bacteria to produce more plastics. So we go into the bacterium, we go into its genome, and we find the enzyme that's responsible for synthesizing the plastic. So where are the genetic instructions that tell it how to make plastic? And we can go out into nature and find lots of different types of these instructions. There's lots of different genes from lots of different organisms that are similar to this that can allow the bacteria to make plastic. And we replace that gene with a new gene from somewhere else in nature. We can also make new types of enzyme as well. We can make new synthetic genes that can allow us to not only make more plastic quicker, but brand new types of plastic as well with different properties. So, we've obviously got a lot of work to do, so we need to go back and do that in the lab. But hopefully I've managed to convince you that bacteria are not all harmful for us. They can do lots of really cool, they can do lots of interesting material things for us. And by researching how to work together with plastics, with microbes and robotics, we can make new materials to hopefully help us have a cleaner planet for the future. Thank you.